All right. Good afternoon, everyone, and welcome to today's webinar sponsored by NAVBO's Education Committee. I'm Amber Stratman, and I will be moderating today's session. We are pleased to welcome our speaker, Dr. Maya Elise Kumar from Stanford University. The title of her talk is Using Single-Cell Approaches to Understand Neointimal Lesion Formation and Growth in Pulmonary Hypertension. I'd also like to welcome Dr. Adam Andreska, also from Stanford University, who will be monitoring today's questions. Before we begin, I'd like to remind you that all NAVBO activities are now easily found on the calendar of events on our website. Please be sure to register for some of these upcoming events. Throughout the presentation, please type your questions into the questions box. These questions will be answered at the end of the presentation. After those questions, provided there is time, attendees will be able to ask any additional questions in real time. If you have a question, raise your hand. You're using the hand icon, and you will be unrecognized to unmute your mic and ask your question at that time. This webinar is being recorded and archived on the NAVBO website for future use. So I'm very pleased and happy to introduce Dr. Kumar today. Dr. Kumar completed her undergraduate education at Williams College and was a Fulbright Scholar in Switzerland before beginning her PhD training at Harvard University in the lab of Dr. Doug Melton. She was a postdoctoral fellow in the lab of Mark Krasno in the Department of Biochemistry at Stanford University, where she applied new methods to probe the behavior of individual cells in the developing lung and used them to understand how healthy airway and blood vessel walls are built and maintained. She is now an assistant professor in the Pediatrics department at Stanford, where her lab studies the pulmonary vasculature. And so we're delighted to have you here today, Dr. Kumar, and please feel free to take it away.
All right. So thank you very much. Thank you to the organizers for this opportunity to present today. I'm delighted to be here. And I'm going to tell you today about a bit about our work on the fascinating problem of artery remodeling that drives disease progression in pulmonary hypertension. And I'll highlight some of the approaches that we've taken using a variety of technologies to begin to unravel what has for many years been a fairly intractable problem. So I hope I can convince you that it's a fascinating problem. So first, a tiny bit of context. My training, as the introduction showed, is as a developmental biologist. I studied embryos. And the question that's always driven me is one of the central questions of developmental biology, which is how are cells coordinated to build tissues? So identifying and characterizing progenitor populations that build a tissue of interest, identifying the regulated steps in the recruitment of those progenitors to a particular lineage, understanding the molecular underpinnings of each of those steps, understanding how all of those disparate events happening in different places in organ development, in this case diagrammed here, the stromal lineages of the developing lung. How are those different events coordinated in order to achieve the proper final pattern? And the system in which my lab studies those questions actually isn't the embryo. It's this very profound artery remodeling that characterizes pulmonary hypertension, which is shown here, comparing a healthy artery, pulmonary artery, to one in the disease pulmonary hypertension. And I hope to convince you that this transition from health to disease can be attacked as a developmental process. It can be interrogated at the single cell level with all the tools of developmental biology. And I'm going to argue that that's a very fruitful way to attack a new type of problem. Okay, so pulmonary hypertension, or PH, it's driven by a progressive remodeling of the pulmonary arteries in which healthy vessels, such as the one here on the left, transition to a highly occluded vessel, such as the one here on the right. So these occluded vessels are surrounded by inflammatory cells. The smooth muscle media, which is the smooth muscle layer bounded between those two darkly stained elastin layers, it's marked, oh good, I hope you can see my pointer. It's marked by these yellow bars in both the healthy and in the PH arteries. That layer expands in disease. And then I think most notably, the lumen is now blocked by the formation of so-called neointimal lesions, which are indicated with this blue bar here. So neointimal lesions, those are the cells filling this whole space. These are cellular accumulations that appear beneath the endothelium, which is now kind of a crunched up cord in the middle of the vessel, and this internal elastic lamina. And these lesions grow and grow and grow, blocking the lumen, increasing the pulmonary vascular resistance that the right heart has to push against, ultimately leading to right heart failure and death. And despite intense study, treatment options for PH remain limited to pulmonary vasodilator therapies, which extend life, but which don't stop this relentless artery remodeling. And what our lab aims to do is to understand the cellular and molecular basis for artery remodeling in PH, with the goal of finding ways to halt lesion progression or ideally even induce lesion regression. So the questions that we want to answer are, what are the steps in this transition from health to disease in the pulmonary arteries? How are those steps coordinated and interdependent? How do progenitor cells contribute to the expansion of these tissues in disease, and how are they regulated? And then which of those events can be targeted pharmacologically in order to prevent disease or ideally reverse it? So answering those questions has been tremendously challenging, because patient tissue is generally available only in end-stage disease. So accessing early disease events with human tissue is very challenging. And then the commonly used animal models of PH, mostly the mouse models, such as chronic hypoxia exposure, those actually don't do a great job of recapitulating the full range of human lesions in PH. They don't make neointima, for instance. And so we set out to make an improved model by reproducing that perivascular inflammation that's seen in PH patients. And we do this by exposing the mice to the allergen house dust mite, which I'll abbreviate as HDM throughout the talk. And we do this over a period of weeks. And so even though the allergen is delivered via the airways, the animals sniff the HDM, the inflammation transitions very rapidly to being centered around the airways, to being centered around the vessels. That happens during the first 2 days of exposure. So following 6 weeks of this chronic perivascular inflammation, pulmonary arteries in the mouse, which is what's shown in the panel on the right here, show robust medial thickening and remarkable human-like neointimal lesion formation with elevated pulmonary pressures and other measures of pH. And I just want to highlight some of the features of these arteries here in this initial stain, because you'll be seeing them again and again in the course of the talk. So what we try to do is highlight the important features of the vessel morphology, which will allow quantitation and analysis, as you'll see in the coming slides. So in pink here and magenta, we're staining for elastin. This allows us to see the boundaries of the smooth muscle layer, and in green is smooth muscle alpha actin, which highlights not only that smooth muscle layer, but also the neointimal cells filling this space here. In red, we have endothelium, and then in blue is DAPI. Okay, so the remodeling in the system follows a very stereotyped and reproducible time course, which experimentally is tremendously valuable. So the medial thickening, the thickening of the media here, happens during the first 2 weeks. Then in the 2 weeks following that, here at the 4-week mark, you can see these small neointimal lesions forming. So these are lesions on the inside of that internal elastic lamina, which I've marked with the dotted line here. And then in the 2 weeks following that, those small lesions have now expanded to fill the vessel lumen to make these really fully occlusive lesions. And this reproducible timing turns out to be very helpful experimentally, as you'll see. So the other thing I'd like to point out is that these cross-sectional views show these neointimal cells as sort of small and round, and that's actually an artifact of it being a two-dimensional section. So neointima is actually, these cells are tremendously long and thin, and they run with the long axis of the artery, which you can see in the schematic below. So these are cross-sections through the short axis of these very, very long cells. And this very distinctive morphology that these neointima cells have in the mouse model matches the distinctive morphology that they have in human patients. So this is a deep tissue staining of arteries from a PH patient sample that's been stained with smooth muscle actin to highlight the neointima. And in these small arteries that are outlined with dotted lines, I hope you can see these long, thin neointima cells that are running along the interior of these vessels. So the reproducible timing of artery remodeling in this system also makes it very straightforward for us to quantitate vessel changes over time from those confocal images that have been stained to highlight the important compartments that I showed you earlier. So here we're measuring medial thickness just in microns, and what we're measuring is the space between those two marked elastic laminae. So it's marked here with the magenta bar in the healthy animal and then in all these schematics. And each dot is an individual measurement from an individual animal, or sorry, this is 3 animals in each time point combined, but each dot is an individual measurement. I want to be clear about that. And what you can see from these plots is that the medial thickening happens all during this first 2-week period and then holds steady after that. And then if you look at neointima, shown here in red, where we're now quantitating just those cells between the endothelium and the internal elastic lamina, again in microns, you can see that neointima formation lags behind medial thickening. It happens here it initiates anyway, between weeks 2 and 4. And then neointima thickens thereafter and becomes more prominent thereafter until by 8 weeks, 97% of artery cross sections will contain neointima. So the ability to clearly visualize the relevant components and compartments of the remodeling arteries that I showed you earlier in those stains has allowed us to make these very precise measurements from confocal images, and then quantitating large numbers of images across multiple animals in this way over the course of artery remodeling allows us to identify when specific events occur, and it gives us a way to precisely identify changes to remodeling dynamics when we start to perturb the system, as we'll do later. For example, in genetically altered mice or where animals are treated with pathway modifying drugs. Okay, so we now have a system for deciphering the cellular and molecular basis of neointimal lesion formation in PH and its expansion in these maturing lesions. And we now have all the tools of mouse genetics available in order for us to try to understand these underlying events that are driving neointimal lesion formation in this system. Okay, so the first question we set out to answer was, where does the neointima come from? It's a very basic question, but the answer was actually, it was very unclear from the current work in the field. And there were 2 competing models in the field. The first is that smooth muscle cells of the media were diving in, breaching that internal elastic lamina and founding the neointima and expanding. And then the second model was that the endothelium was undergoing an endothelial to mesenchymal transition, generating a fibroblast-like cell that was the neointima. And that's how the neointima was forming. And actually, I think this model is probably the dominant model in the field at the moment. So these models are actually easily distinguishable using cell type specific lineage tracing in the mouse. So here, what we're using is an ACTA2 CRE-ER to mark the smooth muscle. And here we're using the GJA5 CRE-ER, which is also called connexin 40, to mark the arterial endothelium. Okay, so in both cases, the CRE-ER is combined with a fluorescent CRE reporter, and healthy adult animals are given a saturating dose of tamoxifen, which should induce recombination. They're then allowed to rest for 2 weeks so that they can fully clear the tamoxifen, and new recombination events have been stopped, and they are then exposed to house dust mite for a period of 8 weeks. So there should be a big, robust neointima at the end of the experiment, after which they're harvested. And then we look at the arteries and we see where has the lineage mark ended up. And the answer is that you only get a lineage labeled neointima. So the lineage label in these images is green. You only get a lineage labeled neointima when you've started with a labeled smooth muscle. So here the ACTA2 lineage trace labels the neointima. However, the GJA5 lineage trace here labels only the endothelium of the resulting arteries. And these endothelial cells have remained endothelium and have not transitioned to a neointimal identity. So smooth muscle is the cell of origin for the neointima in this system. Okay, so smooth muscle cells contain the progenitors of neointima. That's great. But do all smooth muscle cells participate equally in this process, or are there specialized populations that drive the expansion of the media or that found the neointima? What are the patterns of cell division and migration that underlie each of these remodeling steps that we've identified? And so what we wanted to be able to do was to be able to distinctively and heritably mark individual smooth muscle cells in the healthy animal so that we can distinguish them and their progeny from each other as the remodeling progresses. And so for that, we wanted a Cree reporter that wasn't a single color reporter, as I've shown you here, where all the outcomes of the recombinations are marked with the same GFP label or the same TD tomato label. We want one where there are multiple possible outcomes from a recombination event. And for that, you need a multicolor Cree reporter, such as the rainbow reporter that I'm diagramming here. This reporter was made in Irv Weissman's lab. And the way that it works is all the cells in the animal prior to Cree activity are GFP marked. And then after Cree recombinase has recombined the locus, those GFP cells turn off GFP and adopt the expression of one of three potential fluorescent proteins. So either cerulean or M-orange or M-cherry. And these are now heritable and stable marks. So cells can't interconvert between the two colors. Once you're blue forever, and your progeny are blue. Okay, so the experiment is as follows. The rainbow reporter is combined with a smooth muscle specific Cree ER. Then healthy adults are given a saturating dose of tamoxifen with the goal of labeling 100% of the smooth muscle cells with a random pick of one of these three colors. So the idea is that you start with a basically salt and pepper distribution of these three colors. It would have to be salt and pepper and some third spice, turmeric. Of these three colors. And then these labeled animals are put through the dust mite protocol, remodeling occurs, and then if the distribution of the colors in the remodeled arteries after these new tissues have been generated remains random or close to random, then that's consistent with the starting vascular smooth muscle cells contributing equally to generating the expanded media in the neointima. Meaning if all the cells in the labeled smooth muscle turn over just once or twice in order to thicken up the media, and then hundreds of cells dive in order to make the neointima, the pattern that you're going to get in the end is going to be random or very close to random. If, however, just selected cells proliferate at each of those stages, so just one cell in an area proliferates a lot in order to thicken the media, while all of its neighbors remain quiescent, then what you'll get is islands of single color beginning to emerge where that one cell populates a local area. Okay, so this is how we hope to be able to distinguish these different cell behaviors. All right, so what do we actually see? All right, so this is the starting point of the experiment. This is an animal that's been labeled with that saturating dose of tamoxifen and has not yet experienced house dust mite. So this is what the arteries look like at the start of the experiment. This is a vibrotome section, a thick vibrotome section that's been cleared. And so you can see the artery outline is highlighted with the dotted lines, and the individual colors are, I hope clearly visible. So now these animals are exposed to dust mite for different periods of time and analyzed to see how the color distribution is arrayed. Okay, so first, for simplicity's sake, I'm just going to show you our conclusions, and then afterwards I'll show you the primary data upon which those conclusions are based. Okay, so after the first 2 weeks, when medial expansion has occurred, what started as a random pattern remains a random pattern. Okay, so it's still essentially random, and I can show you the quantitation by which we make this assessment. So this is consistent with all or close to all of the cells in the media dividing a small number of times in order to thicken the media. Okay, then in the next step, the seeding of the neointima. Again, and I hope you can see this in the schematic, here are these little tiny neointimal lesions forming. Again, here we see multiple colors in each of these little tiny nascent lesions, meaning that multiple cells have invaded in a local area to start a lesion. Okay, so here's the data. This is, again, a vibrotome section. This is a longitudinal view of an artery after 3 weeks of dust mite exposure. And I think you can see that these medial cells, these are the cells that are oriented around the artery. These cells are remaining, the distribution of colors in the media remains random. But then, and then underneath those cells, these medial cells that run across the artery are the ones that run parallel with the artery. And I hope you can see them here. There's one little cluster here, there's another little cluster here, And then there's another kind of longer cluster that extends along here. And I've highlighted each of these neointimal cells with a colored arrowhead to show which color it is. So each of these little clusters contains all three recombination colors, meaning that multiple individual smooth muscle cells dove in locally to found each of these clusters. Okay, so what happens next is actually, I think, kind of incredible. So during that period of rapid expansion, where you go from just this single layer, patchy neointimal coverage to a fully occluded lesion, this happens in just 2 weeks, fully occluded vessel rather. During that period of rapid expansion, we see bundles of single color neointima cells form. So they stay coherent, they're not migrating a lot, and they're dividing a tremendous amount. And this is consistent with a monoclonal expansion of a very small number of founder cells, potentially just these few little founder cells that we saw at the 3-week time point. So here, let me show you what that looks like. So this is, again, a thick section with kind of an oblique cut through a remodeled artery. And I hope you can see these bundles of single color cells. So here's an M. cherry expressing cluster, one M. orange cluster, another M. orange cluster, and a cerulean cluster, which are have expanded to fill the lesion. Okay, so this tells us several important things. First, that the division of just a handful of cells is responsible for making these giant occlusive lesions. So if we can understand how those cells are selected and what drives their proliferation, that's obviously of great interest. And secondly, and relatedly, it suggests that the neointima doesn't expand by the continued recruitment of new cells diving in from the media continually to bulk up the neointima. It suggests that there's an initial group of founder cells which are selected, which join the neointima, and then essentially expand in isolation thereafter. So that bottleneck, where that small number of founder cells is selected and then the door closes after them, made us wonder whether there's some sort of pre-pattern that's present in the healthy smooth muscle layer that determines which of the many vascular smooth muscle cells will be selected to form the neointima. And if that pre-pattern exists, can we identify those neointimal progenitors ahead of time in the healthy animal? Okay, so essentially what we were looking for is what's diagrammed here in the schematic. We were looking for a gene that marked a fairly rare vascular smooth muscle population that was present in the healthy animals, where when we lineage traced that rare population, those rare cells gave rise to the bulk of the neointima. So that's what we're looking for. And to do this, to try to identify this gene, we turned to single-cell RNA sequencing. We had a data set that we generated from healthy airway and vascular smooth muscle lineages, which are shown here in this T-SNE, where each gray dot is an individual cell. The vascular smooth muscle cluster is circled here. And in red, I'm showing you the transcription of the gene Notch3. So Notch3 is a well-known marker of vascular smooth muscle cells. It's known to be essential in VSMC maturation, and it's known to be upregulated in pulmonary hypertension. So we were already very interested in it. But what really piqued our interest is that if you look at the distribution of Notch3 transcription in this cluster, you can see it's broadly expressed throughout the cluster, and yet it's expressed at radically different levels. So there are relatively few cells that are very high transcribers of Notch3, and there are plenty of cells that are low or undetectable levels of Notch3 expression. So we confirmed this heterogeneous expression that we saw in the transcriptional data set in situ by RNA-scope hybridization, and also using the Notch3 Cre-ER mouse, which I'm showing you here, combined with the TD tomato Cre reporter. So this is a mouse in which Cre-ER is being driven by the endogenous Notch3 regulatory elements. And with a single large dose of tamoxifen and with no dust mite exposure, about 15% of the vascular smooth muscle cells are labeled, shown here in red. And when these labeled animals are then put through the dust mite protocol and make neointima, now the majority of their neointima, which you can see here in these long, skinny neointimal fibers, now the majority of the neointima, 80%, is lineage labeled in these animals. So this is consistent with the Notch3 marked minority population present in the healthy vascular smooth muscle, being the cell of origin for neointimal lesions in this inflammatory model. And this is true both at the population level, which I'm showing you here, and also at the single cell level. So here we're making clones using that same rainbow reporter that I showed you earlier. So now, rather than giving a large saturating dose of tamoxifen with the goal of labeling all the smooth muscle cells, we're giving very limiting doses of tamoxifen so that only rare and widely spaced cells in these healthy animals are marked at the start of the experiment. So when we make clones in this way with a CRE-ER that's driven by a general smooth muscle promoter, such as MYH11 CRE-ER, shown here in these panels on the left, in the resulting clones after neointima formation, only 19% of clones contain neointima cells, whereas when we make the starting clones in the Notch3 population in the healthy animals and allow them to make neointima, now of those clones, now 90% of those clones contain neointima, meaning that these Notch3 cells are strongly biased to generate neointima. Okay, so is Notch3 in this system simply acting as a marker here, or are these cells actually using the Notch pathway in order to accomplish the various stages of artery remodeling? So to test this, we took advantage of the stereotyped timing of remodeling in this model, and that I told you about before, where you have these very defined windows of what happens in the arteries at particular times. And we blocked Notch signaling during 3 2-week windows using the gamma secretase inhibitor DBZ, which, in the interest of clarity, is not a specific Notch3 inhibitor. It's blocking the activity of all the Notch ligands. Okay, receptors rather, sorry. So in this first 2 weeks, the 3 2-week windows are during the period of medial thickening here during the first 2 weeks, then during the period of neointimal establishment during weeks 2 through 4, and then during the period of neointimal expansion during weeks 4 through 6, okay? And so comparing Notch blockade with vehicle-exposed animals harvested at each of these 3 time points marked with the arrow, we could identify a clear difference in only one step. And that step is when neointimal founder cells are recruited from the media to found the neointima. So here, let me show you what we see. So here are those 3 windows again. These are the animals that were harvested at the end of medial thickening, at the end of neointimal lesion establishment, and at the end of lesion expansion. The top row are the vehicle-exposed animals, and the bottom row are the Notch blockade. And I think you can see that in Notch blockade, the media expands just fine. And when when Notch is blocked, you can still expand your neointimal lesions just fine. What you can't do is form new neointimal lesions. So here in the Notch blockade, you see animals are completely devoid of neointimal lesions, whereas in the control animals exposed to vehicle, you see these nascent lesions beginning to form. So it's required during a single step and not during either of the proliferative steps before or after. Oh, and something I should mention is that when you measure the right ventricular systolic pressure in these animals, there's a significant drop when you block neointima formation. Not surprisingly, the animals are much healthier. Okay, so just to summarize, what I've told you is that we have an inflammation-driven mouse model that recapitulates many features of the human disease, and that in this model, smooth muscle generates both the thickened media and the neointimal lesions, and that within the smooth muscle, it's a subset of smooth muscle cells that are marked by high levels of Notch3 expression that are the major cell of origin for the neointima, and that Notch activity is required for that fate switch from a vascular smooth muscle cell to a neointimal cell, but not for either of the proliferative steps, not for medial thickening or for neointimal expansion, and that blocking the establishment of the neointima unsurprisingly significantly improves disease in these animals. Okay, so in the next few minutes, I'd like to just very quickly highlight how we're using this system that we've established to attack 2 really important, outstanding questions. This is kind of like a preview of coming attractions section. So the first is, what does control neointimal growth? We need to identify neointimal growth signals in order to understand how lesion growth is controlled and ideally try to reverse it. We know it's not Notch. What is it? And so the approach that we've taken here stemmed from the observation that, like in the healthy vascular smooth muscle, the neointima is both molecularly and behaviorally heterogeneous. It's not a monolith. And the first observation we made was that proliferation in the neointima during that period of rapid expansion is dominated by the neointima cells that are directly contacting an endothelial cell. So this is a Ki67 stain with Ki67 in red, and you can see that the neointima with red nuclei are very strongly biased to being in direct contact with the endothelium. Here around here, here's the one exception out here close to the median. And we then went on to identify genes that mark those endothelial adjacent neointima cells and to then lineage trace them. So here you can see the initial labeling marks just rare cells within these nascent lesions. And here we're labeling, sorry, not at the beginning of the experiment here in the healthy animal, but rather after 4 weeks of exposure, where now lesions are established but still small. This is when we're giving tamoxifen, and then we're harvesting 2 weeks later here at the 6-week mark. And you can see at the 6-week mark, this initial labeled minority population has now generated the bulk of the neointima after this period of expansion. And the fact that the proliferating cells are so preferentially found in close contact with the endothelium to us suggests that signaling between endothelium and neointima may be a key driver of neointimal proliferation. And we wanted to obviously understand the molecules that were mediating that interaction. And so for that, we turned to single-cell RNA sequencing again. And here we're generating libraries from both healthy and PH mice, and we're including a broad array of tissues. We're including all the smooth muscle lineages, neointima, all the endothelial lineages, immune and fibroblast lineages. And Leah Steffes, who did this work, took great care annotating these cell clusters, validating them by picking, identifying transcripts from the clusters, and then locating those transcripts back in intact tissue by in situ hybridization, as we're showing here with identifying transcripts for the neointima cluster highlighted by this red box, which you can then see identifies the neointima tissue back in the vessels. So Leah's data set represents the first comprehensive genomic description of neointima cells, and digging into the gene expression in the neointima has been very interesting and has turned up some real surprises. And if you'll allow me, I'm just going to take a 2-minute aside to tell you one kind of interesting and surprising story that came out of this analysis. So when Leah looked into the neointima cluster and was trying to validate which of these clusters really represents neointima, one thing that surprised her when she looked at the cluster that she was quite sure was neointima from this tissue localization is that it didn't express one of the key markers that people use for identifying neointima in tissue, which is von Willebrand factor. This is used very frequently as a neointimal marker in antibody stains, and we found that its transcription was not at all detected in our neointimal cluster, and that led us to investigate further because we were very confident we were looking at the right cells. So von Willebrand has always seemed to me like a less than ideal marker because it's a secreted protein that's both long-lived and matrix binding. And as you might have noticed from the elastin stains that I've been showing you all along, there's tons of matrix found within the neointimal lesions. It's prominently expressed by the adjacent endothelium, and it's secreted in settings of endothelial damage or dysfunction, including pulmonary hypertension. So we took a closer look, and here in this these left-hand panels, I'm showing you that von Willebrand protein, which is shown in red here, is readily detected in the neointimal lesions of animal models of PH, human PH patient samples, and even atherosclerotic lesions in the systemic vasculature. You can see there's plenty of red protein stain throughout the neointima. But if you look at where the von Willebrand gene is actually transcribed in these same tissues here in a wide array of animal models of PH in different PH patient samples, and again in those atherosclerotic lesions, it's exclusively transcribed within the endothelium. So von Willebrand are the red dots, and you can see they're co-localized with Claudin 5, an endothelial marker, and they're specifically absent, or they're definitely absent from the neointima that surrounds. They're only found within the endothelium. And so this is consistent with a model in which von Willebrand factor is exclusively produced by endothelial cells, which matches what we see in the transcriptional data sets, that the protein is secreted in settings of endothelial damage and dysfunction in disease, and it then binds to the abundant ECM that's found around it in the neighboring neointimal cells. So this is just kind of a cautionary tale of kind of questioning your tools, and it's a plea to not use von Willebrand as a definitive marker of neointima moving forward. Okay, but this is not actually what we set out to use our transcriptional data sets for. What we were most interested in using our data sets for was to identify the secreted signals being received by neointima cells over the course of artery remodeling. And so to do this in a thorough and unbiased way, Adam Andruska took Leah's data sets and he built these very clever interaction maps with them, which I'm showing you here. So the way he does this is very logical. He makes lists of genes that are differentially expressed between the healthy and the pH cells within each of the cell types that Leah annotated. And then he filters those lists for ligands and receptors, and then maps out the ligand producing sending cells here along the top of the diagram, and then connects them with the cell clusters that are expressing the cognate receptors of those secreted molecules expressed by the top cells. So from this overall map, we can subset out interactions of interest, such as signals from the arterial endothelium that are being received here by the proliferating neointima. So this is now a tractable list. It's a list of candidates where you can test where the expression is, both in mouse tissue and in patient tissue, where top priority candidates can then be functionally tested in vivo for a role in modulating neointimal growth. And just to give you a quick taste of what those experiments look like, here is the result of in vivo testing for one of Leah's top candidates. And again, we're using that staged pharmacology approach that I told you about before with our notch inhibition. So the windows that were exposing these animals to drug are slightly different. We're doing an exposure during the period where neointima is established, during the period where it expands, and then this is a later period than one that I've discussed before. This is after the lesions are fully mature. They're just maintained. If you keep the inflammation going, these lesions will be maintained and will stay large over many weeks. So here we're checking whether lesion maintenance will be altered if pathway activity is changed. And in the plot below, on the y-axis is neointimal lesion thickness. And in the colored boxes below each time point, I'm showing you the percent of arteries that have neointima. And so in blue are arteries from vehicle-exposed animals, and in red are arteries from drug-treated animals. And you can see that at each stage, these paired vehicle and drug-treated animals, at each stage, the lesions are both smaller and less numerous following drug treatment. And that's true even in these very late exposures, where these animals began with fully occluded lesions and fully occluded vessels, very large lesions. And following just 2 weeks of drug, these lesions are both smaller and there are many more neointima-free arteries. So these are very exciting results that we're really interested in pursuing with really gold standard genetic tests. Okay, so another major question that we've set out to tackle is identifying the inflammatory drivers of artery remodeling and neointimal lesion formation. As you can see from the schematic, inflammation is required for all the steps in this process, and we really don't understand how it's accomplishing what it's doing. And this is something that Adam Andruska is tremendously interested in. And so what Adam did as just an initial starting point was to try to understand whether the inflammation was dynamic, which cell populations were present around arteries at different times, and could he correlate those changes in the inflammatory populations with changes in what was happening in the vessels. So first, what Adam did was use broad lineage markers on a time course of HDM tissue sections, and he was able to show in this this isn't Adam's, this is my horrible little plot summarizing Adam's very subtle work. But what happens is that there's an initial wave of macrophage expansion that's very strong and very rapid. And then following that, there's this very steady increase in the number of T cells in this perivascular space. And then following that, at a significant lag, there's an expansion of B cells. And Adam also noted that the inflammation coalesces into these tight spheres of highly proliferative cells that are around arteries and veins. These are marked with these arrowheads here in this picture. And that if you look more carefully at these spheres, they're actually fully formed tertiary lymphoid follicles. So they have a B cell core, a T cell periphery, and if you look at the core, it's interlaced with this beautiful, delicate network of these follicular dendritic cells. So these are really legitimate lymphoid follicles. But Adam wasn't satisfied with those sort of general cell type identifications. He wanted to more precisely characterize the changing inflammatory populations that are in and around the arteries as each of these stages of artery remodeling was occurring. And so he turned to spatial immunoprofiling technique called Codex. So Codex is essentially multiplexed immunostaining in which a single tissue section is incubated with dozens of barcoded primary antibodies that are then sequentially imaged, resulting in a CyTOF level cell type identification while maintaining spatial resolution. So these multiplexed images are then segmented into individual cells, which can then be analyzed using the standard CyTOF pipelines, generating these kinds of T-SNE clusters like what I'm showing you here in C. And then because each cell in these plots is still associated with its original XY position back in the tissue, you can map these cell clusters back into histologic space, which is what's being shown in D. And so what this lets Adam do is quantitate with great precision which lineages are present within signaling distance in and around arteries at specific stages of remodeling. And through this kind of analysis, Adam found that the development of those T-cell rich lymphoid follicles tightly correlates with the development of the neointima. And this focused his attention on T cells, which allowed him to return to his interaction maps where he can pull out the T cell derived signals here, see which clusters are receiving those signals. And as you can see, it's a very long list of clusters that are receiving the signals. And then he can summarize, he's beautifully summarized over here, which cells are receiving the signals. And so you can see that some of these T cell signals are being received directly by the vascular compartment. Here it's being received by smooth muscle cells, neointima, endothelium. But some are being received by the B cells inside the follicles, some are being received by peripheral macrophages, and these are presumably coordinating the overall inflammatory response. So he's able to sort of triage these interaction networks, finding the ones of highest interest, validating those expression patterns both in mouse tissue and in patients, and then using our in vivo model is able to test the outcome of manipulating each of these pathways and disturbing these signaling relationships in vivo in this quantitative way over time. So we're finding some really interesting outcomes here. Okay, so I hope I haven't been too long. I tried to summarize each of our conclusions as I went along. So rather than summing up our conclusions, I kind of wanted to sum up just the approaches that we have found fruitful and the way that we've attacked what to us was a very new problem and how we tried to establish an expandable system, a system that was a strong foundation. And the things that we found fruitful are first to map the key steps and the cell dynamics of the system in vivo using lineage tracing, mapping single cell behaviors, both with that multicolor labeling and then with true clonal labeling, using spatially resolved immunoprofiling to really understand the nature of the cells that are around at key events in the remodeling process. Then the other thing is that we found that in our situation, heterogeneity really matters. In PH, these cryptic populations act as key progenitors at important stages in the remodeling process, and we would have completely missed that if we had remained at the population level, even with the sensitive analyses that we began with. So single cell labeling is tremendously valuable. And then the transcriptional correlate of that is that single-cell transcriptional analysis, single-cell RNA sequencing, making those very powerful interaction maps. That's been a tremendously valuable data set for us in identifying signals and their targets in this unbiased genome-wide way in both health and disease. And this has really been a very valuable data set that we're continuing to mine. And then finally, having a functional in vivo assay where you can modulate elements of the pathways that you've identified through the single cell analysis, modulate them in the dynamic process that you're trying to study and see what the outcome is. That again, for us, has been tremendously valuable and is yeah, something that we're just returning to again and again. Okay, so with that, I'd like to thank the people who did the work, most notably Leah Steffes, who really pioneered the development of the dust mite system and has really pushed forward with understanding how the neointima expansion is occurring. And then also Adam Andruska, who is co-mentored by Etta Speaker-Cotter in the adult pulmonary division here at Stanford. Adam has really pioneered a lot of these in silico methods and has been just an incredible like careful thinker about what the inflammation might actually be doing. He's really been exploring a lot of very subtle and very, very cool analyses that I'm excited to present in the future. And so with that, I'd be happy to take any questions and yeah.
Wonderful, really beautiful, beautiful work. There are several questions in the chat and in the questions box, and I'd encourage anyone else to post their questions there. And I'm going to turn this over to Dr. Andruska, who's going to kind of read you the questions and moderate this part of the session.
Okay.
Okay, I'll just kind of go in order of receiving them. So Joyce Bishop asks, in the time course, the sections in the 6-week time point show some extra VE cadherin lumens in the neointima.
Yeah, oh, good catch.
She asks, does this come from smooth muscle cells? Can the rainbow experiment and pattern reveal more? And she thinks.
Oh, that's such a good question. And truly, I don't have the answer to the last portion of your question. But if you, let me go back so you can actually see what we're talking. Oh, okay. Sorry, this is going to take me eternity to get all the way back. So what we do see occasionally is something that's been described in, holy moly, what's been described in the human as recanalization. So this is the formation of new, hello, where are you? There we go. Here we go. So this little area that I've marked with the, that I've marked with the asterisk here. This is a small, small patent endothelial tube that kind of perforates the neointimal lesions. These are described in people. It's not clear to me whether it's kind of like a pressure release valve. They do seem to connect out to the surrounding capillaries or outside the vessels. So maybe this is a way of kind of bypassing the lesion in a way, but this is something that's totally observed in pH as well as in the model. And about lineage tracing, no, these do not come from the ACTA2 lineage trace cells. These come from the GJA5 lineage trace cells. So these are cells that originate as endothelium and remain endothelium. They just remodel in response to presumably pressure cues, and we'd love to know what they remodel in response to. The question about how would clonal analysis inform our understanding of how they form and remodel during this process, I could not agree with you more. Those mice are in the mouse room and we're just waiting to have the bandwidth to do those clonal experiments. But yes, we absolutely want to see what how these recanalization sprouts are, how they're related to each other at a single cell level. It's a great question. Thank you.
Okay, Steven Spurgeon asks, BMPR2 is the most common cause of hereditary PH, and how does BMPR2 signaling play into the Notch3 model?
Oh, yeah, I don't know. I really don't know. So just at a simple level, we've used BMPR2 mutant mice and put them through the protocol just to see if they would remodel in some way differently, whether the kinetics were different relative to wild type mice. We haven't done it with an enormous number of animals, but so far we've seen no difference. So at least in our system, it's not obvious how alterations in BMPR2 signaling is playing into the processes that we've described. It's also a little unclear to me exactly how the BMP signaling pathway and the Notch signaling pathway would be interacting in this system. It may be as simple as having altered BMPR2 levels changes how an endothelial cell responds to damage, responds to stress from surrounding inflammation, and that BMPR2 cells, endothelial cells, would be more prone to be damaged, and that damage is then propagating the inflammatory signal that's driving the process. But truly, I have only hand-wavy arguments. We do not have any concrete data that links the two pathways. Again, a very good question.
Great. Next question is from Raphael. He asks something we're interested in is why the smooth muscle cells are orienting themselves longitudinally along the flow axis instead of circularly.
Yeah, I don't know. This is turning out to be a theme. So presumably, so the endothelial cells, if you look at healthy endothelial cells responding to flow, they also orient themselves in response to the flow. That's certainly true in intact, healthy vessels. If you look at them, their long axis is oriented with the long axis of the arteries. Presumably, that is then being carried over and transmitted to the neointimal cells that are underlying those oriented endothelial cells. We have not tried to change the flow parameters. Actually, that's not specifically true, what I just said. We have changed. I don't know. I have to think about that. But I haven't checked whether you can change the endothelial polarization and how that would affect the neointimal cell orientation. But yes, we're very interested in how they establish this crazy morphology. They're extremely long, extremely thin, and they're changing their axis as they dive in.
Great. Another question from Zilong Zheng. What evidence do we have for monoclonal expansion in the neointima? And then the second part of this is how does hypobaric hypoxia induce neointima in the mouse in this model?
So what's the second question? I'm sorry, I just want to make sure.
About the effect of hypoxia.
Hypoxia, and how does it interact with the dust mite model? Okay, so let me answer that one. Oh, I took the slide out. Shoot, sorry. So they act synergistically is the short answer. So if you, in the interest of trying to trim this down to a manageable length, I took out the physiology slides. So if you expose an animal to 4 weeks of hypoxia, you get pressures kind of in the mid to upper 30s millimeters of mercury. You get similar pressures for animals exposed to dust mite in isolation, mid to upper 30s. If you combine hypoxia exposure with dust mite exposure, now you have pressures that are up in the 50s and 60s. Presumably that is because they act through distinct and overlapping mechanisms. So you get the muscularization and the expansion of the neointima with the dust mite, and then with hypoxia, you get the addition of a very significant vasoconstriction. That's my hypothesis. I need to test whether truly it is vasoconstriction, but you get an additive effect. And then the first question was about monoclonal expansion in the neointima. Okay, so the reason that we think there's this monoclonal expansion is because when you label cells, here, come on, one more, there we go. When you label, when you have kind of a random label at the beginning where there's many smooth muscle cells labeled with a mix of the three colors, after neointimal expansion, the neointima, which is composed of, I haven't counted in this specific example, but this is probably, 100 or more cells, or more than 100 cells, in this cross section. Oh, what have I done? In this cross section, those cells are organized into bundles of single color cells, meaning that these red cells all came from a starting red cell that became a neointima cell and then divided in situ. These orange cells are related to a starting orange cell that became a neointima cell and then divided in situ. Does that make sense? That's our argument for monoclonal expansion. And that's borne out by the Notch 3 clones that I showed later. Let me see if I can get ahead to those. Here they are. So you can see in these clone images, these Notch 3 clones, this big blue mass, this all came from a single blue Notch 3 labeled cell at the start of the experiment. So this is a big monoclonal expansion where there's very limited cell migration. The cells that are related to each other divide and they stay as kind of just like a bundle of sticks that's occluding the lumen.
Great. I'm going to try to maybe group a couple together, and it's kind of about ligands.
Okay.
First is the ligands being received by Notch 3. If we can talk a little bit about the expression and distribution of that from the data we have. And then also maybe an overall, there's a couple questions about in the interactome analysis, kind of inside out or outside in signaling leading to neointimal formation, if we can opine on that. And then like what connections are between the tertiary lymphoid structures to the smooth muscle cells. Hopefully I didn't muddy that too much. But
okay. So about what ligands or the Notch receptors seeing, it's presumably JAG1. JAG1's expressed in the endothelium. It's the most prominently expressed endothelial Notch ligand that we found. It's, obviously important in many aspects of vascular development. And I think it may be as simple as the Notch 3 cells are able to see the JAG1 more than the lower expressing Notch 3 cells surrounding them. And then when you get, and I didn't fully go into this in the interest of time, but after the medial expansion, one of the features of both human pH and what we see in the mouse is a real degradation of those elastic laminae that normally bound and contain the smooth muscle cells and separate them from the underlying endothelium. So there are breaches of that elastin layer. It's degraded, it's got fragments. And so presumably there's just an opportunity for endothelial cells to signal more with their with the Notch 3 high neighbors in the vascular smooth muscle, and so they are the ones, those Notch 3 ones are the ones that see that signal and can respond to it and migrate in through those breaches in the elastic laminae. Okay, so that's what were the other ones? I've already forgotten. So what are the signaling axes between the tertiary lymphoid follicles and the smooth muscle? It's just ongoing work, and I think Adam would murder me if I told you. So we're working on it as hard as we can, and we'll report it as quickly as possible. As soon as we have solid data, we'll take it to a meeting. And what were the other questions, Adam? I'm sorry, I've already forgotten.
no, I think that kind of focuses on, there's a question from Mitchell Colbank about sex differences, estrogen, progesterone signaling, how this might influence the inflammatory cascade. What do we know so far?
What we know so far is that mice of both male and female mice do this, and they seem to do it with very similar kinetics as far as we can tell. There are slight differences in the timing if you look at different inbred lines, or if you look at mixed mice on a mixed background, which many of our lineage labeling animals are on a mixed background. We don't have like a perfect BELB-C background or a perfect FAB background, a perfect Black 6, but so you can introduce variability there. So genetics does definitely play a part, but we have not identified a sex bias for altering the kinetics of remodeling. And there's certainly no strict requirement for being either male or female in order to do this. About the actual signaling at a molecular level, I just have to confess we haven't looked into it. It hasn't jumped out at us from our analyses of the interactome, but we haven't specifically gone looking for it. So it's a good question and something we should do.
Perfect. Well, I think we're right at 1. If there are extra questions, Bernadette and Sharon will email them to Dr. Kumar and she can answer them directly. We hope everyone enjoyed today's webinar. Look for more information about future events on the NAVBO website. And of course, we hope everyone is able to join us in October at the International Vascular Biology meeting. And hopefully we'll see you there. And thank you again for a lovely talk today. It was quite beautiful.
Oh, my pleasure. Oh, what have I done? Okay. Am I still sharing? I'm not, right?
I don't think so.
Okay, good. Thank you, Amber. That was really
Yeah,
kind.
thank you. Great to finally put a face with a name.
I know. It was nice to meet you. All right.